This is part 404 on chapter 7, looking at the nutrition, ecology, and growth of microorganisms. So just to review so far, the first part you looked at microbial nutrition, looking at how organisms get their carbon from different sources and their energy from different sources, and then nutrition in general, so the different types of nutritional molecules they need. Got that done. The second part looked at transport, so how things are transported across the cell membrane, and we also looked at osmosis, so that's important in there. And then in the third part, we just looked at environmental factors that can influence microorganism growth. So we looked at temperature, what else was there? The oxygen, pH, osmotic pressure, and just atmosphere pressure. So the really important ones are the temperature, oxygen requirements, and the pH for those. So in this last part, it's going to be a little bit longer. We're looking at ecological associations, so how organisms interact with each other, so the ecology part of it. And we're going to look at this again more in chapter 26, which is why chapter 26 is in this unit, along with 7 and 8. And then we're going to look at the study of microbial growth, so how people and microbiologists figure out the growth rate of microbes and look at how the environmental factors can influence their growth rate, so how they actually study these things. So first off, we're going to look at ecological associations among the microorganisms. So these are when organisms interact with each other or they interact with other organisms. So there's symbiotic and non-symbiotic. So symbiotic relationships are where these organisms, they live in a close nutritional relationship, and that relationship's required by one or both of those members in order to survive. And that doesn't necessarily mean the relationship is good or bad, it's just that it has a requirement. So this association is required for something, some reason. So we're going to look at mutualism, commensalism, and parasitism. There's also non-symbiotic relationships. So these are where organisms are usually just free living, they're fine on their own. They may form these relationships, but these relationships are not required for survival. And there's two types, so synergism and antagonism. So again, it doesn't mean that the relationship is good or bad, it's just, it happens, it's not required. So we're going to look at symbiotic relationships first. So these are where those two organisms, they live together in a close partnership. It's required by one or both of those organisms. So we have mutualism. Mutualism is when that relationship is good for both of the organisms. So mutualism, both organisms in the relationship get a benefit. So it's a really good relationship. Commensalism, this is the one where one organism benefits, the other one doesn't really care. It's kind of just going along with it. So, and then the other one, parasitism, this is where the parasite it's good, parasite's getting food and all it needs, whereas the host that's being parasitized is harmed. So that's a bad symbiotic relationship. Well, good for one, bad for the other. So symbiotic doesn't necessarily mean it's good or bad, it just means that there's a close relationship that's required by one or both of the two members in that relationship. The non-symbiotic relationships, these are where the organisms are, again, free living, the relationship's not required. Sometimes these associations or relationships just kind of happen on their own. So synergism is good. Synergism is when the two organisms actually help each other. They don't need each other, but they can help each other do things. So that's the little circle over here on the right-hand side. So we have a nitrogen fixer, which is the blue bacteria. So this nitrogen fixer, it fixes nitrogen, which means that it takes atmospheric nitrogen and puts it into a form that's usable for other organisms. So this nitrogen fixer, it fixes nitrogen into a usable form. Then we have this yellow cellulose degrader, which uses that nitrogen to break down cellulose into glucose. and then that glucose can be used by the nitrogen fixer. So these two bacteria, the nitrogen fixer and the cellulose degrader, they don't have to have each other around, but it's nice if they do. It helps everything grow faster and just get along better. Another type of non-symbiotic relationship is antagonism. This is the bad non-symbiotic relationship. And a good example is antibiosis. And this is how penicillin was discovered. So it's a good story. So there was a plate of bacteria, and you can see on this plate we have bacteria colonies are labeled A up at the top. This plate just happened to be contaminated by a mold called Penicillium notatum. And you can see that's labeled as C on the bottom of the plate. This mold actually produces an antibiotic, and they noticed that there was this clearing of the plate designated by the B in the picture. So this mold produced an antibiotic and actually killed off the bacteria colonies that were too close to the mold. So this is an antagonism. So the bacteria and the fungi, they can live separately by themselves, they're fine. But if you put them together, that mold produces an antibiotic that's going to kill the bacteria that's in the same area. So again, that's antagonism. It's bad for one or both of those organisms in the relationship. So these relationships, we looked at relationships between different microorganisms so far. There's also interrelationships between microbes and humans. And we'll get into this more in one of the next units, I think the very last unit. But the human body, it's a very rich habitat for symbiotic bacteria. We also have fungi and a few protozoans. And this symbiotic relationship, it's usually a mutualism or a commensalism relationship. If it's that kind of relationship, they're just called our normal flora. So there's the normal bacteria and fungi and protozoans that live in our skin or in our intestines. So we also, besides having that normal flora in those mutualistic or commensal relationships, you can also have them turn into parasitic or a type of synergistic relationship. So we have all these microbes interacting on our skin. It's kind of gross to think about, but they're there everywhere. A lot of them are helpful, except when you get sick, then you have the obviously not helpful flora on your body. So one important interaction between humans and microorganisms are called biofilms. So biofilms result when these microorganisms attach to a substrate using an extracellular matrix, so like carbohydrates or proteins outside the cell, they use it to stick together to each other and to a substrate. And this binds them together into really complex organized layers. And biofilms are found everywhere. So organisms don't just live on their own, they live in these biofilms and these complex layers. A really good example of a biofilm is a plaque on teeth. So plaque is actually a biofilm that builds up with the sugars from the food you eat and the bacteria and the fungi that you have in your mouth. So these biofilms are really dominant on a lot of natural environments on the earth. So again, they're found everywhere, including on our own bodies. So these microorganisms and these biofilms, they can actually secrete chemicals where they can communicate and cooperate with each other to help form the biofilms. It's called quorum sensing. I'm not going to get into the details of it. If you're interested, you can read about it in the book or look up more information on it. So here's an example of a biofilm formation and a little bit on the quorum sensing, very little tiny bit. So you have bacteria cells that attach to a substrate. They secrete the sticky substance, which you can kind of see as a clearish blue sticky substance in the picture. Once those bacteria settle down, they can also secrete other molecules. So like the blue circles are called an inducer molecule. The green kind of jelly bean ones, these are quorum-dependent proteins. So they send out signals to other bacteria that they should come join this biofilm. and make it thicker and better. So that's the quorum sensing, or the communication that can occur. So just to kind of review, so we talked about nutrition, transport, environmental factors in previous sections, and then we just went through the ecological associations. And again, we'll look at this more in chapter 26 to kind of get into a little bit more. So last part is looking at the study of microbial growth. So in the study of microbial growth occurs at two levels. So there's growth at the cellular level, where the cell just increases in size. This is not really what we're referring to when we refer to microbial growth. So when we talk about growth, we usually are looking at the second type, which is the increase in population size. So this is where we have one bacteria, it splits into two, those two splits into four, and so on. So the division of bacteria cells, it occurs mainly through a process called binary fission. So first, the parent cell enlarges, so it goes through growth of increasing in size. And then we get into the binary fission part. So that cell, it grows in size, it's going to duplicate its chromosome, so it duplicates its genetic material. It then forms a central transverse septum. So basically it just forms like a divider down the middle of the cell. And that divider, when it's completely formed, it's going to split the cell into two new daughter cells. In binary fission, it's kind of a form of asexual reproduction, so you take one cell, it splits into two identical cells. So this is the actual process shown here. So you have a really young cell at an early phase of the cycle up at the top, and then as that cell grows, it enlarges its cell wall, its cell membrane, it enlarges the volume, and then it also is going to duplicate its chromosome. And you can see that, so we start off with a pink circular chromosome, and you can see it's duplicating, so we have like a blue one that's showing up as well. And that's where that transverse septum starts to grow. So you can see it growing in the middle of the cell from the two, bottom and the top. So as that transverse septum grows, it keeps growing until it splits those two cells in half. One cell has the pink chromosome, the other one has the blue chromosome that was duplicated earlier, and then eventually the two cells actually physically separate from each other. So this is that binary fission process. So that binary fission process, we can actually calculate the rate of population growth. So the rate is the time, or we're going to look at the time required for a complete binary fission cycle. Is called a generation cycle or a doubling time. And this population growth rate is actually exponential growth. So each new binary fission cycle, the population is going to grow by a factor of 2, and that's a type of exponential growth. So a little bit of math coming in here, sorry. So basically that's, you start with one cell, that one cell divides into 2, those 2 cells divide into 4, 4 divide into 8, 16, 32, so you get that exponential growth. The generation or doubling time, it can vary from a few minutes to a few days. So I know E. coli has a generation or doubling time of 20 minutes. So every 20 minutes, that population goes through a binary fission. So you start off with one cell, 20 minutes later you have 2 cells, and then another 20 minutes you have 4 cells, and so on. So it can grow pretty fast. And some bacteria take less than 20 minutes, some bacteria take a really long time to double. It just depends on the species and how they've adapted to that different environmental condition they're placed into. And this generation time, you can actually use it to identify bacteria species. So if you have a bacteria, you find out the generation time is 20 minutes, you can probably narrow it down to a few different species of bacteria, including the E. coli. So this population growth rate, this is the equation for calculating the population size. So it's just an exponential equation here. So again, sorry, a little bit of math. So f and n, or n of f, the function of n, is the total number of cells in the population. So this is what you're trying to figure out. And I is the starting number of cells. So if you start with 1 cell, you put 1. If you start with 2 cells, you put 2, and so on. The exponent, the n, donates the generation time, so what generation we're on, and then 2 to the n, number of cells in that generation. So basically you just take, so if you start with 1 cell, and you want to know how many there's going to be after 2 generations, so after 2 binary fission cycles, you take 1 times 2 to the 2. And 2 to the 2 is 4. 4 times 1 is 4. Wait, yeah, so you'll have 4 cells. Because you start off with 1, the first generation time you'll have 2, the second generation time you'll have 4 cells. So hopefully that makes sense. So besides doing it that way, you can also do what's called a viable plate count, where you can track or figure out the generation time. So if you start with a flask, you inoculate it with a bacteria. And then after 1 hour, you take a sample from that flask and put it on a plate. And then you do that every 60 minutes until you go for 10 hours. So really long time. You just go back every hour, plate a sample from your flask. Then you can grow your plates, and then you can count the number of colonies that grew. So at 60 minutes we had no colonies. At 120 minutes, so 2 hours, we had 2 colonies. 3 hours we had 4 colonies, and so on. So you can see it's kind of every 60 minutes the number of bacteria is doubling over time. And then you can just put this on a graph, so you put time, number of bacteria colonies, and you get this exponential growth curve. So this exponential growth curve, it doesn't last forever, so think about if bacteria just keep on doubling and doubling and doubling, eventually a species is going to cover everything. So eventually you're going to have cell death, or your population has to go at some point. So in laboratory studies, populations typically display this predictable pattern over time called a growth curve. So stages in the normal growth curve that they see when they do this plating of your bacteria over time. So you start with a lag phase, and this is a flat period of adjustment. You have cells enlarging, they don't really grow, they don't really reproduce during the lag phase. So if you look on the graph, we have hours going across the bottom, and then the number of cells going up the y-axis. So you start off with very few cells, you have this lag phase, the cells don't double very fast. It's kind of flat but growing a little bit. Then the second phase is the exponential growth phase. This is where you have that exponential growth we just talked about. So this is a period of maximum growth, and that phase continues as long as the cells have access to nutrients and they're in their favorable environment, so they have the right temperature, they have the right pH, the right oxygen requirements. So you have exponential growth. And because it's biology, eventually the environment's going to become unfavorable, the temperature, usually you run out of nutrients. That's when you get into the third stage in the middle, so you get this plateau of growth. And it's called the stationary phase. The rate of cell growth equals the rate of cell death. That's because the nutrients are becoming depleted, the oxygen's becoming depleted. You also have excretion of waste that's going to cause the environment to not be optimal anymore. So the stationary phase, you have cells dying and cells growing, so it kind of evens out over time. And then if cells are in a container, eventually you're going to get into the fourth phase, which is the death phase. So your limiting factors intensify, you're really running out of food, there's a lot of waste and crap in the tube. So the cells are going to start to die exponentially or very fast. And eventually all the cells will die if you leave them in that culture for as long as possible. So other methods to analyze population growth, so you can do like the plating one, where you just plate them every hour, every 20 minutes, depending on the bacteria. You can also use a turbidometry, which is simple. It's basically you look at how cloudy the growth is. So I'll show you this in lab, hopefully, but in sterile broth, it's really clear. Broth that has been inoculated, it's cloudy. And you can actually measure the degree of cloudiness by how much light passes through that tube. So the cloudiness, the turbidity, the more cloudy it is, the more bacteria you have in that broth. So the more cloudy it is, the higher population size. The more clear the tube, the smaller your population. So you can actually measure the cloudiness as a way of measuring population growth. Another cool way is that you can have a little counter that can count bacteria. So you can do a direct cell count, where you just take a sample. You can either put it on a slide and then count them manually. That's a viable direct cell count. Or you can have this automated system where you just take a sample, you put it into the automated machine, and it counts the bacteria for you. So that's this chapter. So again, we talked about nutrition, different ways organisms get energy, different ways organisms get their carbon. We talked about cell transport, so how cells actually transport these things in and out of the cell. We looked at environmental factors, so again, temperature, pH, oxygen requirements are the big ones. Those ecological associations, which we'll come back to again in chapter 26. And then how we study that microbial growth.